Hello, and welcome to the National Association of Plant Breeders webinar series, How to Breed New Plant Varieties, Imagining and Engineering Crops. My name is Shelby Ellison, and I'll be serving as your host for the webinar. Today's presentation will be given by Professor Kevin Murphy from the Department of Crop and Soil Sciences at Washington State University. Dr. Murphy leads the Barley and Alternative Crop Breeding Program at WSU. A major goal of his research group is to increase the genetic and biodiversity of cropping systems across Washington state through the development of new cultivars and ecologically rooted production practices. Today, Dr. Murphy will be speaking about quinoa production and breeding in his presentation, Keen on Quinoa Breeding. Now I'm going to switch control over of the screen to our speaker, Dr. Murphy.
Great. Thank you so much, Shelby, and thanks to David Francis for inviting me to give this webinar. And I really appreciate everyone who's been able to attend. I have given one webinar on quinoa breeding in the past, about 1.5 years ago, but we've learned quite a bit since then, so it's this one's a little bit different. Okay, so these are the topics I'd like to discuss today. Really, part one will be an introduction to quinoa, a little bit about the origin, history, and then a brief agronomic and nutritional description as well. And then part two, I'll briefly go into quinoa botany and genetics. And I'll try and keep those two relatively short. You can ask me questions on them later, and then spend most of the webinar on our quinoa breeding program here at Washington State University. What are our hybridization and selection strategies, and then what traits we're really interested in selecting for. Okay, so part one, introduction to quinoa, origin, history, and brief description. So quinoa was, quinoa was domesticated near Lake Titicaca in Peru and Bolivia. You can see the arrow there, about 7,000 years ago. From there, it spread north and south along primarily the Inca Trail, which is usually, or in the heyday, about 21 feet wide and stretched from Santiago to Quito. But it stretched all the way up into Colombia and then down to about central Chile. And it arrived, you can see at the tip of the arrow there, about 2,700 years ago. So after the conquest of the Inca Empire, the cultivation of quinoa was discouraged, primarily because of the religious affiliation with many of the indigenous people with quinoa. And so quinoa and other native crops were replaced primarily with wheat and barley. So quinoa cultivation then was reduced to really marginal environments and isolated areas, so very harsh areas of low input farming practices, and that's really shaped a lot of what germplasm we have to work with now, and it actually has had some positive impact on our breeding strategies and then on worldwide adaptation as well. So there are 5 major groups of quinoa, and within each of these, there are dozens to hundreds of different varieties or farmer land races, populations, if you will, breeding for, well, I guess, so farmer selection going on for the full 7,000 plus years. However, more formalized academic type breeding has only been going on even in South America since about the 1970s, so it's fairly recent. These are the 5 major types. There's sea level, south central Chile, with an altitude of less than 500 meters. And for most of what we're interested in, these are the varieties that work for us here in the northern parts of the U.S. And then we also have Andean valleys, so a little bit higher elevation than sea level, but still not necessarily too high. But then it can go up to 4,000 meters. We have subtropical, Solaris, so very salt-tolerant types come out of here, and then the Altiplano, so very high elevation, 35 to 4,000 meters in elevation. And so at WSU in August of 2013, so last August, we hosted a International Quinoa Research Symposium here. So we had about 150 people attend from 25 different countries. And the talks were all recorded. So throughout this presentation, I think I have 4 or 5 different links that I'll share with you on where you can find topics of specific interest. And so here, if you're interested in these groups and really genetic diversity of quinoa worldwide, Didier Bazile from France has linked seminar here, and you can get to it from this link. Most of the topics I'm talking about today I won't be able to go into too much depth, so I'll add these links as we go. So quinoa is thought to be very broadly adapted. It's grown currently in major regions over a very wide range of latitudes. So 2 degrees north to about 42 degrees south in South America, so that's a range of over 3,000 miles. And then quite a bit now you're seeing in Europe and Canada, Denmark, the US, Pakistan, India, Australia, China, so all over the world. We grew our first crop in the Philippines last year. It grows very wide range in altitude, so from sea level to 14,000 feet. The idea that it only grows in high mountain areas is a myth. It can grow very well at sea level as well. It grows in a very wide range of annual precipitation, so about 6 inches to over 110 inches. We have all of those rainfall zones represented here in Washington, so we're selecting in pretty much all of that. We don't go up to 110 quite, but close. So in general, quinoa is very drought tolerant. It also grows in a wide range of pH soils, and it's also frost tolerant. And that the frost tolerance really depends on the stage of the plant. It's more frost tolerant during the early vegetative stages, and once it starts to flower, it becomes less frost tolerant, only to maybe 26 degrees Fahrenheit, but that's usually not an issue when it's flowering. So again, here's another excellent webinar done by Senator Jake on what is the global potential of quinoa, with the link below. He was our keynote speaker for the symposium. So one of the reasons quinoa has become so popular in the last 20 plus years is its nutritional value. So imports into the US from about 4 million pounds per year. That was in 2007, and last year there were 73 million pounds imported into the US. Worldwide, there's about 105 hectares grown, Bolivia and Peru. So back to the nutritional value. It's an excellent source of protein. Most common range is 12 to 18%. 16% is about average. We've seen anywhere from 7 to 2% reported in the literature. But it's really the balance of the amino acids, the 10 essential amino acids within quinoa seeds, that make it a complete protein and really make it a valuable source of protein for humans. It does have high concentrations of calcium, magnesium, iron, copper, and zinc. It's very rich in beta-carotene and also rich in niacin, riboflavin, and several vitamins. It's high in essential fatty acids, and in particular linoleic acid. It's due to the high starch content. I'll maybe discuss a little bit, or if you have questions, we have a graduate student working on this now. It can be used very similarly to cereals and flour production. And so we're also testing different varieties for how they are used in processing. So we work with a food scientist who extrudes quinoa and different varieties to see how they work. And then one of the interesting parts is that of quinoa is it is gluten-free. So for people with celiac disease, it's an excellent alternative to wheat. And even many people with just gluten allergies, this is a good option as well. So one of the most interesting and potentially useful aspects of quinoa is its salinity tolerance. And we do have some salinity issues in Washington. There's even more in the western U.S. A few years ago, I was doing a hop research trial, and we had to just pretty much give up the whole hop trial because the soil was too saline. That was in the Yakima Valley. And we're seeing higher and higher soil salinity within that valley in Washington State. So quinoa is a potential good option for farmers to grow where saline soils exist. Saline soils are defined anything 4 decimils per meter or higher. At that stage, most crops start to see a significant yield decrease. Quinoa, on the other hand, doesn't lose any yield up until about 20 decisiemens per meter, and then it starts to see some hit from yield. From 20 to 40 decisiemens per meter, quinoa can yield about 60% of normal, which is quite good. And it's even been shown to produce seed when grown in seawater, which is about 55 decisiemens per meter. This is a picture of wheat grown in a saline soil about 4 decisiemens per meter. And so you can see how damaged wheat is by the salinity. Just at 4 decisiemens per meter. Barley can go up to about 6 decisiemens per meter before it starts seeing some issues. And so quinoa, again, can go up to about 20, so 3 to 5 times what wheat and barley can handle. Okay, so moving on to part 2, quinoa botany and genetics. So quinoa, Chenopodium quinoa, is a member of the Amaranthaceae family, and it's grown primarily for its seed, but many people do also eat the leaves. We actually thin quinoa every year in our breeding program quite a bit, so we always have quinoa leaves to eat, and they're quite good. I enjoy them, but I also enjoy pretty much any kind of green, from chard and kale to nettle. So, but it's a great green, and one of the interesting things we've noticed is that it'll keep for quite a long time, 6 to 8 weeks in our refrigerator without showing any damage. So that's pretty different than lettuce or many of the other greens. It's a pseudo-cereal, so not a true grain. However, we still often call it a grain in our program. We had 5 Andean farmers come visit last August, and they told us that, having quinoa being called a pseudo-cereal is insulting to them, and they prefer it being called a Andean grain. So we still stick to grain. It is related to beets and spinach, and also to common lamb's quarter, so Chenopodium album and Chenopodium berlanderii. It's gynomonoecious, so that means it has female and perfect flowers on the same individual plant, and I'll show a picture of that here. This is a photo taken by Adam Peterson, a graduate student who just finished in my lab. And so this shows, on the left, a hermaphrodite flower. You can see this. The sepals have been removed, so you can see the anthers here. And then right here, it's a little hard to see here, but this is only a female flower. So there are no anthers in this flower. And this is quite common. We will often see hermaphroditic flowers on the very ends of these little spikes and panicles, and the female flowers will be more located proximal to the central stem. Quinoa crosses at a rate of 10 to 17%. There are reports of outcrossing even higher than that. So it's still predominantly self-pollinating, but it can outcross quite readily. It's wind-pollinated. So for isolation distances, we often, if we need to keep things separate, we keep it at about 300 feet. And at that point, there is less than 0.1% of any crossing to occur. Through most of our trials, though, we just keep it a couple feet apart from each other, and it doesn't seem to cross too much there. But within each variety or population, they're very diverse. They're almost, if you would compare them to wheat, it'd look more like a heterogeneous landrace. We see very few quinoa varieties that are actually quite uniform. So there is a lot of crossing going on, and that's not necessarily a bad thing. We try and work with that as much as possible. So the flowers are grouped together to form a primary panicle, depending on how closely the the plants are together, that'll determine how much branching there is on each quinoa plant, but they have the potential to branch quite significantly. And in many parts of Bolivia or Peru, they often do multiple harvests. So they'll cut the primary panicle first, come back a week or 2 weeks later, and then start cutting branches and getting more seed. One of the things we're breeding for is mechanical harvest cultivation. So we do seed a little denser and try and get the seed to mature all at the same time. So again, quinoa is facultative autogamous annual. So that's essentially it's an inbreeder with a capacity to outcross. It's an allopolyploid, so it comes from 2 different genetic backgrounds as opposed to an autopolyploid. The base chromosome number is 9, so it's 2n4x equals 36. Luckily, the inheritance is primarily disomic. That's true for most of the qualitative traits. We do see some tetrasomic segregation in a few qualitative traits, and then in quantitative traits as well. And then down here, Jeff Mon, he's from Brigham Young University. He's put together a great webinar and genomic resources for Chenopodium quinoa. And the researchers, Jeff and Rick Jelen and others at Brigham Young University, have been studying quinoa genetics for 15, 20 years, so that they're really a phenomenal resource for us here in the U.S. So there are some related species. I'll just touch on 2 of the most important ones because we see them quite a bit in our fields. The first is Chenopodium album, common lamb's quarters. It's an allohexaploid. It is considered a weed in most of the U.S., and it lacks domestication traits that are found in quinoa, such as shattering and a hard seed coat. So that essentially means that lamb's quarters or Chenopodium album has dormancy that most quinoa varieties do not. In fact, almost all quinoa varieties do not. So lamb's quarter, for example, 3% of the seeds will germinate right away after the seed shatters and hits the ground. The other 97% has various densities and thicknesses of the seed coat, so they'll keep germinating over the next month to decades. Quinoa doesn't have that, so that was one of our main questions when we started the breeding program was, is quinoa going to become a weed in other fields? And there's really very little dormancy. There's only one variety that we found that has any dormancy at all. We tested it down in Oregon with a grower down there, and it had about 3 months dormancy, and that came out of Argentina. But almost every single other quinoa variety we've seen will germinate quite immediately. So when you do combine it and the seed goes blowing out the back of the combine, as some will always do, it won't become a weed. So it'll germinate right away, and then that seed will die over the winter, at least in the northern U.S., and then and that'll be the end of it. Okay, so another related species, this one native to the U.S., is Chenopodium berlanderii. It's also called horizontle or Nuttall's goosefoot. Horizontle is the term used in Mexico and primarily for when it's used as a leafy green. This berlanderii is an allotetraploid, and so it has one diploid ancestor in common with quinoa. It is quite widespread all the way up to Alaska from Mexico, and there is evidence of domestication in the eastern U.S. Its seeds have been found in these rock shelters in Kentucky, and this is one of the species we've made some wide crosses to. So we've crossed quinoa with berlanderii with the hopes of being able to transfer some heat tolerance into quinoa. So quinoa, if it gets, just a rule of thumb, over 95 degrees during pollination in the summer, the pollen will become sterile, and so no seeds will be formed. Well, most of the U.S. that's a problem because it does become quite hot. So you can either beat that by planting early or maybe even planting late, so you miss that period, or one strategy we're working on is breeding for heat tolerance in quinoa, and And to do that, we've made crosses with Chenopodium berlanderi. And so, again, down here, Rick Jellin from Brigham Young University has put together, this is a fantastic webinar on related species to quinoa. Really great, worth looking into. Okay, so we'll move on to part 3. We're going to talk about our breeding program. So start with some hybridization first. When our WSU quinoa breeding program started in 2010, and when we started, it was with a grant from the Organic Farming Research Foundation for about $15,000. That really helped launch quinoa research at WSU. Back then, there was just one person working on quinoa. Now we've got 10 different faculty and 10 grad students, all working on different aspects of quinoa, not just breeding, but a lot of agronomy, entomology, food science as well. So it's really exciting. We're a nice tight-knit group. We work well together. We also work with Oregon State researchers there and Utah State University quite closely, in addition to Rick and Jeff from BYU. So we had to start somewhere. I'll talk about some populations we got from Rick and Jeff at BYU, but first we wanted to select our own, populations, really our own progeny, our parents, which one we did several years of trials, seeing which varieties did the best here, and then throughout we've been making crosses with them. And so compared to wheat, barley, several other crops, quinoa is extremely hard to hybridize. I won't go into too much. There is some tricks to doing that. We're in the process of putting out an extension bulletin on quinoa hybridization. But so here you can see just the initial steps from far left are taking off the leaves one by one, and then just isolating a few of the flower clusters here, and then finally we put it in this tubing, dialysis tubing for isolation, and wait for it to flower. Just as it's starting to flower here, you can see the clusters. They're about ready for emasculation right now. These white lines here show about where we cut each flower cluster, and what that does is allow us to emasculate. So it exposes the anthers and allows us to remove them. As I mentioned before, some of the flowers are female, so if we do see a female flower and don't have to emasculate, we'll automatically go for that. It's much easier. However, that really depends on variety, depends on the environment. There's a lot of things that trigger the formation of females versus perfect flowers, and so this is often what we have to do, is emasculate. And then after emasculate, we'll just, again, in the dialysis tubing, put the female parent and the male parent together and just tap it for a few days, and we get pretty good success from this hybridization here. We do this all through a microscope. So again, wheat and barley we just do with our naked eye out in the field or in the greenhouse. This we have to do entirely under a microscope so we can see what we're doing. So, and then we have to verify to make sure the cross actually happened and there wasn't any selfing. So what we often do is just look for dominant morphological markers, and so what we've done entirely so far is just do crosses with seeds or seed coats with different colors, or leaves with different colors, or big differences in plant height or leaf shapes, anything like that, and that we know the genetic background for, and then we can make sure that there has been a cross. So this picture shows Temuco here, this yellow flowering variety on the left, and Bio Bio, this red flowering variety in the middle here. And then these are two F1 progeny that show, it's a little hard to see in this picture, but they're more pink than they are red, so it's more of an intermediate type. But because the F1 is large, the red seed coat color is largely dominant, we do see this red color here. Again, there's over about I think about 60 different colors of quinoa that have been reported. So it's hard to say, you know, dominant recessive for each one of those, but we can come pretty close with our estimations. And then we'll take the population. So we'll first of all, we'll grow out the F2 in the greenhouse, really just to increase the seed in a safe environment, so we don't want to lose any of that precious seed. So we'll increase it in the greenhouse, and then we'll grow the F3 to F5 populations out in the field. And so we're doing this in two mega environments of Washington State, the west side, so west side of the Cascades, and the east side of the Cascades. And the west side of the Cascades is much cooler, much wetter than the east side. The east side is hot and dry, and so they're very different. And so we're already, even though we don't do much selection at this stage, we do some random selection, but just the impact of natural selection is already affecting these populations. For example, last year on the west side of the Cascades, it rained quite a bit in August and September, about 4 to 5 inches. It's usually pretty dry over there, and so we had a lot of pre-harvest sprouting, and I'll show a picture of that later on. But the sprouting selection pressure was fantastic. We lost about 80% of our lines because due to pre-harvest sprouting. So the 20% we saved, we're really hoping that we can get some good sprout tolerance lines out of that with consistent sprout tolerance. And we think we can. One of the best parents for sprout tolerance is a variety called Redhead, which was bred and selected by Frank Morton of Wild Garden Seeds. And he selected that one after two very, very wet August and September fall harvest seasons. So we'll grow those out in two diverse environments, and then we'll start some advanced yield locations. So again, these are our three primary locations. This is on the Olympic Peninsula. We work with two farmers here. They get about 30 to 50 inches of rain a year, depending on which location they are, and definitely on the year. It can be more or less than that. And then, so again, this is where we test for sprouting tolerance, mold tolerance, that type of thing, but very little heat tolerance. So this whole area of Western Washington is ideal for growing quinoa based on, you know, kind of cool summer temperatures. We also are doing selection for heat tolerance here in the lower Yakima Valley. We actually do some heat tolerance selection here in Idaho as well, Lewiston, Idaho, where last year it got up to 107 degrees, and in our acre of breeding plots, we didn't get one single seed. So that it was a little too hot to get any good selection pressure there. But we're going to keep trying. We are looking at irrigation and the effects of irrigation on heat tolerance as well. And then here in the Palouse, we've got about 1.5 million to 2 million acres of dryland wheat and barley production right now, some legumes, lentils, chickpeas as well. And this is where we do the bulk of our research. So Pullman is located here, and we've got trial farms all around this area where we're looking at dryland quinoa production and seeing how quinoa will fit into a common rotation on the Palouse, whether it would come after winter wheat or where it fits in, what it would replace in the rotation, or if it can just diversify rotation. One of the main groups of farmers we're working with too is a no-till or direct seed group of farmers. So it depends on the county. Whitman County, where we are, it's about 20 to 30% no-till growers. Down here, southeast, Walla Walla County, it's about 80% no-till growers. And so there are different traits that we're selecting within the no-till farming systems as well. In general, these are our traits of interest. They go beyond this. So nutritional value, that's kind of a broad one, but we have many different traits we're looking within nutritional value. Grain yields, very important. And then also early maturity, that's extremely important for us. Most of the varieties that we test, if they come from one of the main subgroups from Bolivia or Peru, they won't mature in time in our climate. So looking for early maturity is extremely important. as is heat tolerance. And then other things we're looking at: seed size, lodging resistance. Some of these lines grow 8 to 10 feet high, which is too high really for us because they start to lodge. So salinity tolerance we look at in particular with BYU and Utah State. Drought tolerance, we have some insects that are a problem: lygus bug, aphids. Whether or not we see genetic resistance to these is still to be determined. Nitrogen use efficiency, saponin content, and sprouting resistance. Down here, this is a picture of Hannah Walters and Adam Peterson, two graduate students who both just completed their master's and are still actually working with quinoa. So this is Downy Mildew. This is the #1 quinoa disease worldwide, a disease of quinoa. You can see the gray and pink discoloration of the leaves. This would be a fairly susceptible variety here. So this is something we're also looking at, primarily on the west side, but we also do see it in eastern Washington too. It does prefer cool, moist climates. You see, again, the discoloration, chlorosis, necrosis. It can drop yields down to 0% if it's severe. And this is something we're working very hard on here. We have seen it, and again, we're working with a lot of South American scientists who are also working on downy mildew resistance. Resistant varieties. This is one resistant variety. This one, again, this is cherry vanilla. This is from Frank Morton of Wild Garden Seeds, and because he's done selection of quinoa in western Oregon near the Willamette Valley for so long, he's already got some good varieties that are fairly resistant to downy mildew. So this is a picture of pre-harvest sprouting. You can see all the little sprouts coming out of the seeds here. This is quite common. We've just out of the last 4 years, this has happened twice to a majority of our seeds on the west side. So this is again genetically controlled, and something we are I think on the cusp of figuring out, and any new varieties we'll release for western Washington might also become applicable to other parts of the U.S. as well, where there are those August summer rains. In addition to genetic resistance pre-harvest sprouting, we're looking at panicle architecture. So this type of panicle here allows a lot more wind to blow through it, and it keeps it dry, and we've noticed that these are often a lot less moldy than these ones here on the right. And it's really our natural tendency just to select these big, beautiful heads here full of seeds. But for western Washington or very rainy areas, these just tend to, the moisture just gets trapped in there, and over time will cause all of these to mold and or sprout. So these are the ones on the right side are better for drier areas, and then these ones on the left side are better for wetter areas. So saponins are really key in determining the processing capacity of quinoa. So I guess probably the major bottleneck we have in the U.S. right now is the lack of a processing facility to remove saponins. They've got really nice state-of-the-art facilities in Peru and Bolivia that do this through a combination of washing and abrasion, light abrasion. But we don't have that in the U.S. yet, and so we know there's a really big market for quinoa. Farmers now are realizing they can grow it with the right varieties, and they're figuring out how. So we, especially in this area, the Palouse, we know we can grow it. But we haven't been able to get that seed to market, and within the last month, we've heard that Andean Naturals, Sergio Nunez de Arco and Don Kukin are putting together a quinoa processing facility in the Sacramento Valley. So for us, for growers up here in Washington, it's about 4 to 7 cents a pound to ship it down to Sacramento Valley and get it clean. So that's going to make a big impact. One of the questions is, though, can we breed saponin-free varieties, and there are a few out there. They're patented. Um, a couple we've seen growing in this region are from the Netherlands. They actually haven't done quite well in this area because I think they're not very heat tolerant, but it's a really interesting idea. So we have some saponin-free varieties or populations that we're developing right now, where we've made crosses and we're testing those out in the field. A problem with a saponin-free variety is that then you lose any benefits that the saponins impart upon the seed, and one of the main ones is that it is a bird deterrent and also has been shown to be an insect deterrent. But then on the other side, you do have a clean quinoa seed without the bitter soapy saponins on the outside. So our goal is to release both saponin-free quinoa varieties and ones with saponins on them as well. And then we'll let the growers figure out what works best for them. I'm not going to talk much about this now. We've got two students. This one here, Geang Wu, and here she's working on texture and micronutrient concentrations of different quinoa varieties. So really interesting research. And then Rocio over here with her PI, Juliana Norato. They're both from Peru but work at WSU. They're working on bioactive components and its effect on obesity in quinoa. So there's a lot of research we're doing there. This is all later stage selection. So once we're growing out 7 of our best lines just for both of these researchers to do some more work on, and if we can, then we'll release varieties that are more nutritious or have better heart healthy compounds or whatnot. There's a lot of different options there. So we are working with Brigham Young too on some quinoa mapping. They've done quite a bit of this, so our role is doing some phenotyping for these projects. We're hoping to identify QTLs for some traits of interest, as well as to develop varieties for our region. We've done a couple years of phenotyping so far. We've looked at 5 different populations, all coming from these 8 different varieties here. I just color-coded them to show you kind of the areas where they come from. The purple ones here are from more of the Altiplano regions, Salares regions of Bolivia and Peru. So very high altitude, salt-tolerant, drought-tolerant, and they also have just very high levels of or differences in downy mildew susceptibility. So those are 3 of our parents. When we grow them here, they don't do well at all. In fact, they don't even make any seed. And then we chose some parents from the Chilean coast as well. So these populations were developed at BYU and grown for about 7 years. So we've been testing the F7 and the F8 generations of these lines. And so these are from the Chilean coast. And when you see some of these, especially from down here, NL6, KU2, they make big, beautiful heads. And if, in good years, we get a nice harvest from them. These are what the parental lines look like when they're grown out in Pullman, so in the Palouse. NL6, these are a few of the other ones, KU2. And then these, so you can see the big head on NL6. When you look down here at Chucapaca, this is really what we get, a nice tall plant with some spindly, like finger type branches coming out. And again, these won't make any seed. They'll start flowering in August, and by then it's too late to actually get a good seed crop for us for these. So these are the mapping populations we have. We don't have any data to share with you on these yet. Hannah just published her thesis, and we're sifting through all that right now to see where we are. But we grew these first 4 populations out 2 years in a row. This is the number of lines we grew out each year. this last year we had a really nice big population from this, we call it our Go population. And this is the only one with a Chilean cross with a Chilean. So we have great hopes that progeny coming from this population will actually be well adapted to our area, and if we're lucky, we might get something we can actually turn into a cultivated variety. So when we phenotype, we're looking at many different traits, but not all of them. Plant height and architecture are very important. Flowering, seed maturity, again, downy mildew. Leaf and inflorescence color, seed color, saponin content, protein content. And this is what we're looking at on almost all the progeny lines. When we get down to a certain point, then we can start looking at the nutritional value and other things that really we can't do on 800 different lines, but we can do on 10 or 20. So here's a picture of the one of the populations, and so here in this lower corner you can see one of the lines. It only grew about 18 inches, had nice big leaves. It might work out great for an edible leaf variety, but it doesn't make seed in our area, so that could be a drawback, but that's fine too. It'll make seeds somewhere. And a lot of these are quite tasty. So right next to it, a sister line. This one grows to be about 9 feet, and again, this one didn't make any seed either. So there's a tremendous amount of genetic diversity within all these. You can see some downy mildew here on this leaf, and so within this is really our pool to select from, and what we're hoping for is something about 4 to 5 feet tall that matures by late August to early September, and is both heat tolerant, drought tolerant, and depending on where we grow them, also with a good potential for downy mildew resistance and pre-harvest sprouting tolerance. So I'll end there, and hopefully we will have time for questions. We again did start this research with funding from Organic Farming Research Foundation. Cliff Bar Family Foundation Seed Matters has provided a very generous fellowship for Hannah Walters. So her entire master's degree was funded through Cliff Bar. WSU, CSANR, they've provided funding as well, and the USDA OREI program has also provided a very generous amount of funding for WSU and Oregon State, Utah State, and BYU as well.